I want to thank the organizers for allowing me to come to your beautiful city. When I told my wife that I was invited, she said, we're going. And we're here. So what I want to do is to give you a sense of how the brain is plastic, because that's, of course, in rehab, what we're trying to take advantage of. Now, the biggest question you're going to have, I know, is where in the world is Lethbridge? And it's where that X is. You can see Calgary. It's in Western Canada. It's in the middle of nowhere in many ways, but it's the Rocky Mountains. And Adele Diamond, who will speak later, is 1,000 kilometers further west in Vancouver. It's a big country. Okay, so the take-home message is that basically all experiences you have, including those that are preconceptual, that is, experiences that your parents had before you were born, before you were conceived, are going to affect brain development for the rest of your life. So if you have problems, you can blame your mother or your father. It's entirely their fault, and I'll do the same. I think this is important as we start to design interventions. It's important for education, and it's important for influencing public policy. So questions that we're going to ask is, how does the brain develop? It's not, when I took my first course on the brain in 1966, the idea that the little brain was anything different than just a small version of the big brain would seem preposterous, and now we know it's totally different. They're different brains, they're different species in a sense. How is this development affected by all the experiences we have? What might the mechanisms be that allow these plastic changes to occur? And how does this have outcomes in children? All right, so the brain develops, and many of you know this, but we'll just start with the metaphor. Basically, you could not have a blueprint for a brain. It's too complicated. You can't have 10 to the 14 connections specified in advance. And so what we're going to use, or what we did use, is what I'll call the Michelangelo Solution, and that is when he was creating the statue of David, he talked about how David emerged from this big piece of stone. And basically, in brain development, it's the same thing. Your brain's going to emerge from a brain that's twice as big in terms of number of cells at birth as it is today. And of course, as Michelangelo was using chisels to knock pieces, okay? I won't start over. As Michelangelo was using chisels to chip away pieces of of stone, we have chisels that are chipping away pieces of brain, and those chisels are hormones, experiences of various kinds, as you'll see. And this will be on the exam. This is the organization of brain development, starting with the birth of cells. The cells migrate to wherever they're going to be, and they basically grow up. But the key thing is item 6, and that is the cell death and pruning. And you can see here, is this going to work? The cursor? Yep. Nope. Be light. You can see the line here. The line here for visual cortex. So we see the number of synapses, and going across from left to right, it's increasing, and at birth it's quite high. It reaches a peak around age 2, and then for visual cortex around age 5 or 6 you start to get rid of synapses. In the prefrontal cortex it's delayed. It doesn't reach a peak until about age 3 to 5. It kind of sits there until early adolescence, and during early adolescence is when we get the change. So you can see this is now just looking at the number of synapses in prefrontal cortex, and you can see 2 lines, one of which is layer 5, the other was layer 3. It doesn't really matter. And you can see there's a peak, and then around age 8 or 10 it starts to drop. And my least favorite group of people in the world are 12-year-old girls, and many of you have been a 12-year-old girl, and you can remember what it was like. Things change very fast. So at the peak of of synaptic loss, you were losing about 200,000 synapses per second. So 200,000 gone, 200,000 gone, 200,000 gone. So you can see why the behavior, and it's more true of girls than boys because it's a little earlier, and the hormones are really coming on quite spectacularly and quickly. The behavior is so changeable. The brain is so changeable. But there's a second time that there's a big change. It doesn't show up in this figure, but it does here. This is a cartoon showing that there is a change in plasticity at maximally around age 14 or so. So a little earlier in girls, a little older in boys. But you can see here that it's starting around 8 or 10 and it reaches that peak. And so one of the things that happens is because there's so much plasticity, the brain can change for good and not good. And if you look at the onset of psychiatric disorders, you can see the common thing here is they're occurring basically there. So around age 8 to 10 to 12, you're seeing these disorders arise. And what's going to be important is that a lot of these, that the pathway, the trajectory to get to these disorders was laid down very early in life. But for example, depression, you're going to see the effects of early experience in the first 2 or 3 years showing up as depression in girls around age 14 to 15. So the seeds are sown early, but it doesn't show up until you get this prefrontal change. All right, so a lot of my studies use lab animals, a lot of them use people as well. But the lab animals, people often say, well, what's this got to do with people? Well, there's various ways we can do this. We can study behavior, and so we've got a rat on the left there who's reaching through a slot to get food. They're very good at this. My colleague Ian Wishaw has spent 40 years inventing tests to look at motor coordination and so on in rats. They're very good at using their digits in spite of what the textbooks, not ours, but other textbooks say. This is not unique to primates. We can look at the structure of cells. So the center there, you can see Golgi stains of a pyramidal neuron. And of course, in Spain, you know about Ramon y Cajal, how much can be learned about the organization of the brain simply by looking at these beautiful creatures. And finally, we can look at epigenetics, so changes in gene expression. And so I think you all understand that all of the cells in your body have the same DNA, but it's not expressed the same way. So the cells that make up your hair and skin and eye and brain and so on all have the same DNA, but they don't show the same characteristics because different genes are turned on or off. And the key thing here is environment can do the same thing. It can turn genes on or off, and that's going to make a big difference to brain development. So if you see here the old nature-nurture distinction, on the left you see nature, nurture on the right, and of course it's neither. It's the meeting point in the center, which is there, which is epigenetics. So, there's a laser here, okay. Yeah, so we see here this meeting point, and the changes in gene expression are going to lead to changes in brain plasticity, which are going to lead to the outcomes that we're all familiar with. Now, what events in brain development are altered by postnatal experiences? Well, we can have, as you can read it as well as I can, changes in the number of neurons that are generated. So there is postnatal generation of neurons. It's specific to certain regions of the brain later in life, but in the first few years, there's quite a bit of neurogenesis remaining. We have synapse formation, of course, as you saw earlier, and then we have, during adolescence, the pruning of cells, especially in adolescence. Okay, what factors are influencing brain development? Sensory and motor experience is obvious, and one of my favorites is tactile stimulation. So you can look here at, we're going to call this kangaroo care, for an obvious reason, but we can see the similarity. So the touch is really important, and skin-to-skin touch is really important. And in South America, children have much more of this skin-to-skin contact than we do in North America or in Europe. And in fact, one of the tragedies for Indigenous peoples in Canada, it's the effect of colonialism brought about by the British, the French, and the Spanish largely. The outcomes, as you may know, have not been good. And in the part of the world that I'm from, because the children were put in what we call residential schools, schools that were designed to make them assimilate and become European, it didn't work. But one of the things they weren't allowed to do is to speak their native languages, and they weren't allowed to touch. And even today, there's very little contact among adults and children, which is clearly not a good thing. So if we look at maternal massage, so we've got massage here, we've got skin to skin, we've got infant massage, and in rats we can do the same thing. So we could use a little paintbrush and we can lay the little baby rats out in a little line like Vienna sausages, and then we can take a little paintbrush and stimulate them. And now we have another way of doing it, and I don't know if you have it in Spain. In Canada, it's called a swifter. It's a duster, basically, and it's about this long with feathers, and we can do the whole bunch at once. So they're just lying there, and we're tactily stimulating them. Or we can stimulate the mom here with a child's hairbrush, and we can brush her. What happens is that the skin is changed by this tactile stimulation. So we're going to see that it affects both brain and behavioral development. We can see this in adults. And so if you look here, you can look at the successive reaching. So here's a rat that's reaching for food. It got tactile stimulation when it was an infant, newborn, and now as an adult, we haven't touched it since. We haven't done anything to that rat. We can see that performance, this is percent correct, is way better in the animals who had that early tactile stimulation. So somehow, the tactile stimulation has got into the brain and changed it. The question is how? How does that happen? How does the skin change the brain? And one of the ways we've discovered is because if you fall and you scrape your hand, what do? You rub it. Why do you rub it? Well, it feels better. Why does it feel better? Well, you're making something. One of the things you're making is called FGF2, fibroblast growth factor 2. And FGF2 has receptors in the skin and in the brain. So when you rub the skin, you're making FGF2, and it goes in the brain and changes the brain, and it's a positive thing. So if you think about children or lab animals with early brain injuries, tactile stimulation is a way into the brain to stimulate synaptic changes. Anything that alters FGF2 should alter brain development, and as you'll see, it does. So many of the treatments that we could initiate are actually affecting the production of FGF2. And that's, as a result, having the same effect as tactile stimulation. Language and music is obvious. It's going to influence brain. I'm not going to say much about this. Adele might say more, I'm not sure. But the main point is that language and cognitive activity, including music, change the trajectory of the developing brain. Now, I want you to look at this figure. This is looking at the development of vocabulary in children from age 1 year, 12 months, to 36 months, and you can see we can put them in 3 groups, or Hart and Risley put them in 3 groups in this initial study 20 years ago. And you can see there are children who have about 300 words and children who have about 1,200 words. So there's a 4-fold difference in the number of words. Now, this turns out to be critical because once you're on the trajectory, here you keep going, and here you keep going. So children who have high number of words at 36 months, if we look at them when they're 11 years old, 8 years later, those children have a larger vocabulary than the mothers of these children. Something's going on here, and you're going to see in a second what that is. But can you change this? So there was a study in New Zealand looking by the education department, looking at the effect of going to school between ages 5 and 14. Did it change any of this? And teachers don't like to hear it, but the answer is no. So they gave tests of reading and mathematics at age 5 and 14, and the children who were at the top in those 2 scores were still at the top. The children who were at the bottom on that low trajectory after 8 years of schooling are still there. So something has to happen earlier in order to get them off that trajectory. Music training also changes the brain. The IQ, it basically, musical training is like another language. So it's changing the brain, and also the earlier, the better. And these effects can be seen throughout life. And in fact, the incidence of Alzheimer's or other dementias is reduced in people who learn to play music as children. And it therefore increases cognitive reserve. Poverty is one of the worst predictors of brain development. Let's go back to this figure. I've now put in brackets SES, socioeconomic status, and you can see that that's what's related. That's in part one of the causes of this difference. Now, the key thing here is that it's not that the people with low SES are not as smart, that their intelligence is lower. It's that they interact with their children in a different way. And one of the ways that's different is the higher SES families tend to have what I'm going to call serve and return language interactions. So pretend this computer is my child, and I look at the child, and the child looks at me, and I say, what do you want to do? And then the child comes back, and we go back and forth like this, like in tennis. We're going back and forth, eye to eye, and we're going to have contact. You see a lot more of that, considerably more of that, in the high SES than in the low SES, where the parents are scrambling to make enough money to get the bills paid and so on. The children are left on their own, and there's not so much of that. And in fact, the difference is 30 million words by age 4. That is, the child has listened to language in the home to 30 million fewer words. Now, obviously, they're not different words. There aren't that many words that kids are going to hear. But just hearing the words over and over again, you could not make up that deficit, ever. And that's going to lead us to that trajectory. And it's not only the words, it's the grammar. The grammar is different as well. So something has to be done in those first 3 or 4 years. If we look at the brain, when these children are now adolescents, one of the things we can see is that the language areas are significantly smaller in children from impoverished families. Now, these results come from the United States, and I'm guessing that the effects in countries like Canada or in Spain are going to be not as dramatic as they are in the US, where the disparities in income is so large. Nonetheless, you can see the areas that are in red and yellow are larger in children from high SES families, and that correlates with their vocabulary. So you can see this vocabulary effect here. It's been replicated now in several studies, so we know it's not a one-off thing. Stress. Stress is huge. And if you look at adults, this is a cartoon, you can see a neuron from the prefrontal cortex. So both Dr. Diamond and I are prefrontal chauvinists. So you hear a lot about prefrontal cortex. And the medial frontal region, so this region here, is the cells become smaller with stress in adulthood, and the cells in the orbital frontal cortex, the stuff behind our eyes, become larger. Now, if we have the stress in adulthood, the older you are, the less change there is with time. So if I'm stressed, unfortunately, I'm in a group that isn't going to change much. If I were 20 again, I could revert back to baseline with enough time. But when children, the effects are quite different. So if we look at preconception stress, so your mother or your father is stressed, and in rats we can do it this way, put animals on an open platform, they sit there for 20 minutes twice a day. In this case, we're doing it for 30 minutes, and with the dads, we're doing it for 27 straight days. So they're getting put on that platform, their corticosterone levels go through the roof, because rats don't like sitting out in open places waiting for a cat to come or a hawk to come to get them. So why do it for 27 days? Because the sperm takes about 30 days to turn over, so that the sperm that the rat has after these 30 days roughly of stress is different. The gene expression is different than it was before. That's how the environment can change how the children of that dad are going to behave. If we look here now at the effects of the stress on, again, I'm using the reaching task for consistency, but you can see here's the control animals, and here are the animals who've had stress. And this is similar to having a prefrontal lesion in the magnitude of the effect. The animals, even with extended training, never make up that deficit. So that they never were stressed, their dad was stressed, then they were conceived. Now we're looking when they're adults, and we can see that. And in fact, if we look at the next generation, Gerlinda Metz in our group in Lethbridge has been doing that, and we've been collaborating with her, same result. So this crosses generations. We're seeing the effect going for some time. One of the big effects is on prefrontal cortex. So we have a reduction in the number of synapses in orbital frontal cortex, which is the opposite of what adult stress does. So you can see the effects on the brain are still there, but now it's very different conceptually. And the effect on the hippocampus is also very large, although the biggest effect is in orbital frontal cortex. There's no change in medial frontal cortex in this case. And in contrast to most stress studies, the effects are bigger in females. Normally it's males that are more affected by stress. In this case, it's females. If we have gestational stress, that is in utero stress, we're going to see outcomes that are similar, and that is we're going to have impaired motor, social, emotional, and cognitive behavior, increased corticosterone levels again, and increased corticosterone levels in reaction to later experiences. So for example, in one study, the stressor we used was noise. We tried to mimic traffic noise, and we looked at the effect of that noise on the mom when she was pregnant. We looked at the effect on her offspring when they were adults. And the effect was corticosteroid levels, the baseline levels were highly elevated in the offspring. They had all sorts of behavioral impairments, and their reaction to stressors, mild stressors, such as being tested in that reaching task, increased the corticosteroid levels even more. So some of the tests, the rats are more anxious, they're less likely to go out in the open. They play differently. I'll come back to play in a second. So the male and female rats, as well as people, have sex-dependent play patterns. And basically, early stress, gestational stress, feminizes males. Females, it's just reduced in the amount. I've already told you about changes in reaching, and we can see changes in cognitive behaviors as well. Now, if we look at what the effects on the brain are, we have smaller brains. So you're stressed in utero. In the case of the rats, it's just the last, it's during the time that the cortical neurons are being produced. So in the case of humans, the second trimester. And we have abnormal connectivity. There's a really interesting study done in Quebec. So in 1998, there was what, up until that time, was the worst natural disaster in Canadian history. It was an ice storm, and I realized that for you, an ice storm is going to be a foreign concept, and that's a lucky thing. So what happens in an ice storm is you have freezing rain, and the freezing rain can produce havoc, and I'll show you right now, and then I'll come back. This is what happened to the power grid. It's gone, and it's you can see all of the ice here. You can see all these power lines are down. Now, this happened in the coldest time of the year. It was in January. The average temperature, it turned out that winter, was minus 20 Celsius, and people had no power for up to 6 weeks. You can imagine there was a fair amount of stress, and people were cold. The whole infrastructure of the society had broken down. So what you could do is you can study the children of women who were pregnant during that ice storm, okay? And you can compare their behavior in their brains to their older siblings who were conceived and born before the ice storm. Same parents, same environment post-storm, it was just during the storm. And what you see here is that there's an IQ drop of about 10 points. They have slow motor development, slow language development. They have slow abnormal play, and this has been now measured at these years. There are MRIs that are being analyzed, but one of the things that has been analyzed is changes in epigenetics. Now, this next slide is going to look complicated. It really isn't. But here's what we're going to do. We're going to take all of the offspring. We're going to give the mums a rating scale. This was done right after the storm as to how stressed they were. Some weren't stressed at all because their power didn't go off. Okay, so they're going to be blue, and then, as you'll see, and then red are the ones who were stressed. So if you look here, here's blue. So these are individual children, and this is the mom's rating scale. So these moms thought it wasn't so bad. The further you go in this direction, the more stressed the women were. Now, if you look at the colors of the genes, here are genes. You can see there's a break right about here. These are green. Green means they're off, and orange or red means they're on. And I hope you can see that what's going on here and what's going on here is very different. So this is at age 15 years, and we're looking at the changes in gene expression related to something these kids experienced in utero, and that was the ice storm. Their mums were stressed. So we can have a long standing effect. And of course, postnatal stress. One of the most interesting studies is the Aversive Childhood Experiences study by Vincent Folletti and his colleagues in the United States. Basically, they now have about 170,000 middle-aged adults, and they retrospectively have given them a scale and asked them if they had any of these early aversive experiences. And aversive experiences, I'll show you examples, but it's what you would expect. And it turns out people are more likely to have them than not. About 50% of the population in their study has at least one. And there's a powerful relationship to health in middle age, so 50 years later. So these are examples. I love this idea here of turning gold into lead, so turning children into lead, basically. And you've got these kinds of things. These are what you would expect as early stressors. What does it do? You can see the kinds of things that it does. 55 decided to jump down here. It should be outcomes after age 55. And you can see all sorts of things. It's not just brain. It's brain in the whole body. Now, one of the big things here that I think is quite interesting is the effects vary with the number of stressful experiences. So you're 3 times more likely to be a smoker if you had 2 of these stressors early in life than if you had none. If we look at suicide, the same sort of thing. Drug addiction is 50 times higher. The incidence, the likelihood of being sexually assaulted if you're a woman by age 50 is 5 times higher if you had been exposed to these early aversive experiences. Now, why would that be? Well, the easiest answer is that who are you most likely to be assaulted by? Somebody that you know. Okay, why would that make a difference? Because your interpretation of the context is not accurate. You don't notice that there's a problem. And what's that related to? The effect of that early stress on prefrontal cortical development. And in addition, there's an effect of early stressors on telomere length, and that's related to the onset of Alzheimer's. And you can see here the telomeres at the end of these genes are getting, or chromosomes, sorry, are getting shorter and shorter. And the more ACEs you have, the faster that loss of telomere length becomes. So, stress summary: stress is a bad thing, and you can have stress preconceptually, in utero, or in infancy, or later in life, and what we see is huge changes on the brain and its plasticity. So basically, plasticity is reduced as a result of these early stressors. I think we can reverse this, and one of the ways is with tactile stimulation. That's one of the things we're trying now. Psychoactive drugs. Every psychoactive drug that we take is designed to change the brain. That's the whole point of it, psychoactive. And it turns out that every psychoactive drug you've taken has left a footprint in your brain. And one of the footprints I'm sorry to tell you about is a drug that many of you took today already. I did, and that's caffeine. Even caffeine will leave a footprint. Now, is this a bad thing or a good thing? I think because I'm a caffeine addict, I'm going to tell you it's a thing. It's not a bad thing. But on the whole, a lot of these changes are not good. So Terry Robinson from Michigan and I initially started these studies almost 20 years ago, looking at changes in the structure of neurons. Nobody had thought to do this before. And this is the cells from an animal who got amphetamine, 14 straight days, and we're looking in the brain 4 months later, an adult rat, 4 months later. And I hope you can see visually that the amphetamine neuron, that is a neuron from an amphetamine-stimulated rat, has more synapses. It's bigger, therefore has more synapses, about 30% more synapses than its littermate who got saline. Now that was true for neurons here. If you look at neurons that are in the orbital frontal cortex, it's the opposite. That is, the cells are simpler. So it's not just that you're growing more synapses, it's that in different regions of the brain you're having drug-dependent effects. And in prefrontal cortex, you see an increase in response to the stimulant. Same thing is true of nicotine, cocaine, methylphenidate, and so on. Now, one of the interesting things is in schizophrenia, the cells in prefrontal cortex and medial prefrontal cortex are simplified. They have fewer synapses. Those of you who have worked with schizophrenics know that virtually all schizophrenics self-medicate with nicotine. Presumably what they're trying to do is to increase the number of synapses in that medial area. What happens in the orbital area? Well, in the orbital area, you've got cells that are larger than they should be, and so the nicotine is going to reduce it. So basically it's working in the 2 different regions in different ways. So what kind of drugs are we talking about? Well, all of these and more. And I think you can see I've got a little plus sign beside the ones that we've looked at, and looking at early exposure, that is gestational exposure, giving the mom the drugs, and then looking at the offspring. Now, we haven't looked at the 2nd generation or the 3rd generation yet, but that's always something to do. So if we look here at nicotine, the mom is exposed to nicotine. The fetus is exposed to nicotine. This is a female fetus, and her reproductive cells are exposed to nicotine. So we're affecting 3 generations at once here. And we see a change in gene expression. Parent-child relations, this is obvious. One of the most important bonding experiences you'll ever have is with your caregiver early in life. And we know that we're going to see the power of this in studies looking at children who have been institutionalized. Now, this is a study looking at children who were adopted into the United States. This is Megan Gunner and her colleagues at Minnesota, and they're looking at the effect of having been adopted into families, and then we're looking now at adolescents. And if you look at children were in 2 groups. They were adopted at a mean age of 12 months, but some were adopted earlier and some were adopted as late as 2 years. The question is, what does it do to the brain? And so they looked at 12 to 14 years. And if you look here on the left, it's prefrontal cortex. So the striped bar is control. This is volume of prefrontal cortex. And you can see it didn't matter. This is early adoption or late adoption. It's smaller. So the age at which you were adopted is affecting the size of your prefrontal cortex. In their study, they're really talking about dorsolateral prefrontal cortex. And if you look at hippocampus, actually now we see a difference where the late adopted kids have a larger effect on volume of hippocampus than the earlier adopted kids, predicting, of course, that they're going to have larger effects on memory and spatial abilities. Other studies have shown reduced IQs and all sorts of cognitive and social-emotional deficits. The effect is reduced if they stay in the same foster home or adopted home for life. If they start moving from home to home, as you can imagine, the effect's going to get worse. Peer relationships. I have the privilege of working with Sergio Pellis at our university. He's arguably the world's expert on play behavior, and he studies play behavior in rodents as well as carnivores, and he's studied about 60 species of monkeys. And basically, if you're a rat or a cat, your job in play is to try and nuzzle the back of the neck of your playmate. So this top guy is trying to nuzzle the back. What's the other guy doing? I don't think so. And they're pushing him away. And what you get is this rolling behavior. And those of you who've watched kittens, that's what you see, this rolling behavior. They're trying to do the same thing. This is not fighting. If you were fighting, if you're a kitten or if you're a rat or any other animal, where's the last place you want your face? By the teeth of the other guy. So fighting is aimed at the butt because there's no teeth there. This is very different. Children with rough and tumble play, the details are a little different. What's important is that there are rules for play. So one of the rules is reciprocity. I get to try, then you get to try. I get to try, you get to try. Some kinds of early experiences remove that reciprocity. And so if you have had early stress, for example, you don't seem to have integrated that rule into your brain, and other animals won't play with you because they say, oh, you're not giving me a turn, I'm not going to play with you. So if we look at the effects, what does play do to the brain? Play changes the way the synaptic loss occurs. So we see the pruning is dependent on the nature of the play. And also, the more play you have early on, the more plastic your brain is later, especially in prefrontal cortex. I won't bore you with all the studies we've done. Just trust me that that is in fact the case. We can manipulate the amount of play. Diet seems obvious. Diet is going to make a difference. You can see the question that Bonnie Kaplan asked, does prenatal micronutrient supplementation improve children's mental development, i.e. brain development? And the simple answer is yes. There's a nice study done in Gambia in East Africa in which the people's diet changes throughout the year. So the dry season and the rainy season has a different diet. And so the question that the investigators asked was, is the brain different because of this? And the first stage in the study was to look at gene expression. So what they did, this is the paper, what they showed was that at the time of conception, whether you're in the rainy period or the dry period, when you look at the gene expression in the infants when they're born, it's different. And this just illustrates the difference here. This is Z-score, basically. And so you can see this difference, which may not look huge, but in fact it is. So genes are turned off and on by the rainy season diet. Another thing that's happening is changes in gut bacteria. Now, there's been a real interest in the last 5 or 6 years in the microbiome, and that is understanding all the bugs in our gut. And you may know that there's more DNA in your gut, in the bacteria, than you have in your body. So you've got all these foreign beasts inside you with DNA, and they can actually change you. So we now know that the bugs can influence the brain in a variety of ways. They can do it directly, or they can do it indirectly. Directly by producing various chemicals which then go to the brain or through the vagal nerve as well. It turns out that there's a new term now, psychobiotics, and the idea here is that these microorganisms in the gut will actually change the brain and change behavior. And if that's true, then maybe you could actually use fecal transplants to change the brain of people. So let's just give you a far-out example that there's a clinical trial on now in Ireland, and that is you have people who are schizophrenic, their microbiome is abnormal, so what if we were to actually give them transplants, fecal transplants? Now obviously you're going to have to be very careful where you get this basically poop from in order to do the fecal transplant, but you can change the microbiome, and in turn the idea is that that'll change the brain and change the behavior, normalize the behavior of these schizophrenics. According to Bryan and Dinnan, the early results were positive with their first trial with schizophrenia, but they're arguing that this may be a whole new class of psychotropic compounds that we can use to alter the brain in a good way, or in a bad way if the microbiome is changed in a bad way. Perinatal injury. Now, I know that one of the things that the Institute does is rehabilitation, and perinatal injury is for children, obviously, has a huge impact on brain development, not just cerebral palsy, but other things as well. And I can tell you as a sort of broad brush here that the outcomes of perinatal injury vary with age at injury, age at assessment, and the behaviors measured as well as sex. Now, age at injury, there was this idea that was known as the Canard Doctrine. It was named by Hans Lucas Teuber, but it was actually incorrect. But what the idea was that earlier is better. So if you're going to have brain damage, have it early. And then you'll show better recovery because the brain's more plastic. That was the logic. It's not true. The worst time to have brain damage is at birth. The brain is not so plastic in response to that. If there's a good time, a better time would be, say, 1 year to 2 years. And in our rat studies, we've been able to show that there are a variety of things that happen. So if you had brain injury in the second trimester, the outcome of that is actually quite good. If you had brain injury in the third trimester, the outcome is really bad. And that's because the brain is at a different stage in development and responds differently to that perturbation. The age at assessment makes a difference, and one of the reasons is, remember the graph I showed you with vocabulary, and you saw the different trajectories. Well, very often the trajectory is being set by the early injury, but when you look, say, at age 3 or 4, you don't see much of an effect of the injury. And so I've been to court many times and been asked, I've assessed children, they've had a prefrontal injury early on, and the judge will say, well, can you predict? And these kids say age 6 or 7, can you predict the outcome of this child? And my answer is no, because we can't see what the effect on the prefrontal cortex is until these kids are 12 or 14. Well, of course, in the justice system, that doesn't work. You can't wait that long in order to settle lawsuits and so on. So the age at assessment becomes a real problem. The behaviors measured is huge. So you're measuring cognitive behaviors, motor behaviors, socioaffective behaviors. On the whole, the cognitive behaviors show better recovery than motor behaviors, and they show better recovery than socioaffective behaviors. So clearly what we need to do is to have interventions early that are focused more on the socioaffective domain than on the cognitive domain, because the cognitive stuff's going to improve anyway. Now obviously it's not going to be normal. So doesn't matter when the brain is damaged early in life, it's always smaller. That's not true in us. In adults, the effect on brain size is trivial. The effect on brain size with early injury is really quite large. And if we look in our rodent studies, if the brain is injured in the first week of life, so equivalent to the third trimester in humans, the brain is reduced in size by about 20%. But in spite of that, if we introduce interventions such as tactile stimulation, the animals recover quite well. So basically what I've said here is that they can do more with less somehow. So it's positive in that sense. But if you don't intervene, of course, the outcome is not very good. So interventions make a big difference. So tactile stimulation in our hands has made a big difference. So we have a perinatal injury, and then we'll tactily stimulate the animals the first 2 weeks of life, and then leave them alone till they're adults, and we see that there's amazing compensation. Rearing animals in complex environments, same story. In fact, if we rear the mom when she's pregnant in a complex environment, and then we damage the brain at birth, the outcome is better. So once again, we're seeing that this prenatal, we've done preconceptual studies as well, we just haven't finished. I'm going to predict that the preconceptual effect will be like the prenatal effect, so we're going to have the mom or the dad live in a complex environment for a month, take them out, and then we mate them, and then we look at the offspring that we give perinatal brain injuries to, and their outcome is better. Okay? The immune system. Many of the proteins that are first found in the immune system, are also expressed in the developing central nervous system. In fact, I've heard it said that basically the immune system, the development of the immune system in evolution preceded development of the brain. I'm not going to go that far. But nonetheless, we know that there's a big impact of the immune system on the developing central nervous system. So if we look here, we've got a pregnant mom. She's exposed to some kind of bug, the flu, or whatever it happens to be. More recently, things like Zika. And we can, Zika virus, you can see it's changing the cytokine production. The cytokine production actually affects the placenta, and it goes through and affects the brain. So we can see a root here of immune compromise as a result of these bacterial infections or viruses. Now, the key thing here, of course, is that we know that certain diseases are related to, such as schizophrenia, to mums having been exposed to various bugs. So we know this isn't preposterous. And in the case of Zika, it's actually now been shown to be pretty straightforward. So we know that Zika is related to changes in cortical development, not just microcephaly, but that's one of the extreme effects. So if you look here, we've got the mum, the infant has fibroblasts, and the precursor cells, the ones that have green here, are affected by the Zika virus, and that changes the production of those neurons. So what you get is something like this. So this is a baby with typical head size. This is microcephaly, and this is severe microcephaly, as we're seeing in Brazil in all those children who were born whose mums were exposed to Zika virus. So where did we come and where did we go? I'm telling you that brain development is complex, brain development is prolonged, and I didn't emphasize the fact, but I will now, and that is, it really isn't finished till around age 30 years. We used to think brain development was done at maybe 2 or 3, and then we adjusted that to say, well, no, not till late adolescence. And now we know, looking at changes in pruning, that pruning, especially in prefrontal cortex, goes on until at least age 30 in males and females, maybe at age 28. So it goes on for a long time. And as a result, you're going to see the effect of experiences on the brain lasting a long time. And I often ask people who have to be older, and you'll see why, when do you think you became who you are? And most people who are in their 40s or 50s will say, women will say, oh, my late 20s, and men will say early 30s is when I became who I am. I would say in my case, early 30s. I was hired as a professor when I was a child, when I was 28, and I'm not that person. I thought I was pretty mature. I mean, I'm assistant professor, big deal. I got a job, and yay. I'm going to stand in front of students and tell them all sorts of wise things, which wasn't very true, but I thought it was. But it's much later than that. It was probably 35 before I could say, oh, that was the guy who was now finally mature. Brain plasticity is influenced by a lot of early experiences. I think the more we study it, the more surprised we are that virtually everything we experience during development is altering brain development and the outcomes later in life. And I think that one of the real insights we've had is that it's not only gestational and perinatal experiences, but preconceptual experiences. So I tell my students, undergraduate students in neuropsychology, I say, now, ladies, I want you to look at this data very carefully, and I want you to observe the effects that you see in your colleagues in the room who are males, and they're out partying, they're going to football games and having a lot to drink and so on. What effect is this having on their gene expression, and how is it going to influence the development of your babies? And the kids just look at me like, what? Are you crazy? I say, no, not really. But these early experiences have profound effects. Okay, now the studies I've talked about obviously weren't all done by me, and there's been lots of help, but I want to point out, this is Fraser Mustard, the late Fraser Mustard, who was a cardiologist, and he had this insight into the idea that early experiences would have an impact on the brain. And so for the last 25 or 30 years of his career, he traveled around the world taking this message, and he started the Canadian Institute for Advanced Research, and one of the programs that he started was one on child brain development, which I've had the privilege to be in. So he's had a huge impact worldwide, especially in Canada, Australia, and Africa. And then the others I can thank as well, and I'll stop there. And I think the questions are held to the end, but I'm not sure if there's time now. Thank you. Thank you for listening.